myasthenia gravis. Have you learned about this neuro yet?
Yeah,
Well, we did neuropathophys, but not.
Neuro. Okay. You've heard of this, probably. Okay. And what happens in myasthenia gravis is that you lose muscle tone, you lose muscle strength, and it's progressive. And it tends to start essentially like a space and before it moves out distance. And persons with myasthenia gravis have circulating antibodies, autoantibodies against receptors for acetylcholine. And you know, acetylcholine is required for a nerve impulse from one nerve to the other nerve, then you get the muscle and other muscle too. So that's one mechanism for muscle weakness, and that's how it shows up in myasthenia gravis. Multiple sclerosis is another neuromuscular disease that has an autoimmune component, but it works by a different mechanism. The autoantibody isn't against the neurotransmitter, it's against the myelin sheath of the neurons themselves. And you know the myelin around the neuron impacts the speed of movement of the impulse along the neuron. You eat away at that thing, and you're no longer able to get the move along as it should. All right. Alright, so we got three pictures, and I like pictures that look like cartoons. I always love cartoons, so it makes sense to me. Okay, so a normal person, in order to get a muscle to contract, acetylcholine has to be released into this postsynaptic space here, out of neuron number 1. So acetylcholine comes in, acetylcholine in this space then meets with the receptor on the muscle to cause communication between nerve and muscle so that the muscle moves. Person with myasthenia gravis, they don't have any trouble making acetylcholine. Acetylcholine is made just fine. In fact, sometimes is made more in a greater volume than these guys over here, thinking that if I just make more of it, I can make this muscle move. The problem is the patient is making antibody against the receptor for acetylcholine. And we learned about how competitors get in the way on enzymes. Well, this is the same kind of an idea. There's a competitor for the acetylcholine. These autoantibodies get in the way. So no matter how much acetylcholine the nerves are making, they can't get where they need to be on the surface, on their receptor on the muscle, so the muscle does move. Okay. you know is what we said already in terms of what it is determined on. We know that viruses can change the structure and how cells of cells look. We didn't talk about that it was a particular virus, and I probably won't ask you if this particular virus causes the same. Immunodeficiency diseases are those that make the immune system not work very well. We kind of talked about this before. We said that there are certain situations, and some are congenital, so they be born like that, where the B cell side of the immune system doesn't function. There are some inheritable disorders where the T cell side of it. There's certain that the complement system doesn't function well because one or more of the proteins is not being produced normally. Phagocytes can be sick. Sometimes it's a kind of a global kind of deficiency. T cells and T cells are non-functional or hypofunctional. So that this individual is going to be very at high risk for certain types of infections because the immune system is not going to be able to either produce antibodies as they should or be able to produce cytotoxic T cells as they should. So the person, the baby, the individual is a warrior with no armor. Now, there are some immunodeficiencies that are acquired. You know about one of those is the one that we always talk about. Number one, not the only one, is what? HIV/AIDS. Right. Born, most people are not born with it. Some are unfortunate enough to be born of a parent who acquired it. But mostly when we think of HIV, acquired immunodeficiency, something bad happens to an immune system that was otherwise normal before it came into contact with this virus. Secondary immune disorder. What might be a secondary immune disorder. We already talked about that kind of today. Is that some people get into such trouble, like a person who has that infected disease, the bone marrow is not a bone marrow that does not function, is not consistent with life. So we have to do something for that person, and sometimes that something is either a bone marrow transplant. In order to do that, we have to make whatever immune system that immunity that the person has or hypoactive or in kind of in a coma, so that they don't reject. Sometimes people have unfortunate occasions to acquire cancer, and they need chemotherapeutic drugs that not only kill the cell line is, but it also impedes the immune cells and their ability to do what they were designed to do. So we can treat one thing and cause a immunodeficiency at the same time. We talked about our primary immunodeficiency or forms of lack of cells, cytokines, or both. If a person is has a B-cell defect, they're going to be the ones who aren't able to make antibodies, not able to make them in titers to be detected. B-cell defect is somebody who's generally is a sinus can't make T-cells, so they're going to have a particular risk for some are destined to be T cells. Those destined to be T cells are matured in the bone marrow. If there is a problem at this area, this person is never going to make any antibodies at all. So the closer the defect is to the stem cell, the more severe the disorder will be in the T cell, if someone is a thymic, they have Bizard syndrome, and they don't make a thymic. So they're going to have terrible, zero T cells. A person who has Bizard syndrome and has no T cells will have a clinical picture that's very similar to what you think when your T cells don't work. Yeah, it's going to be very, very similar to AIDS, because AIDS, that's the cell line that virus attacks. And little by little by little, the T cell numbers go down and down and down and down to a critical level to where now you're having clinical symptoms of AIDS. So George, no thymus, going to have symptoms very similar to AIDS. All right, so we can make a thymus. We got past that point, but you're not able to make certain enzymes like ADA, adenosine deficiency. That means the T cells don't work properly. You have some, but they don't work efficiently, so we're going to still be more susceptible to what kinds of infections are you more susceptible to without T cells that work properly? Protozoans, and viruses, and fungi. Those things that go in the cells, change the character of the outside the cells, wake up CD8 and CD8 destroy the infected cells. Okay, so we got past this thing. So we got to the bone marrow, we made the T cells, but we have some problem with the T cells that they aren't able to make the structure of the antibodies either don't work, or they don't make enough of them. They're hypoglobulinemia. So people who have T cell issues are going to have more problems with the current bacterial infection, because antibodies are what does most of the work in clearing the blood and the system bacteria. The T cells are what does most of the work in getting the body of viruses and protozoans and fungi. Here's style. Here's the end of the cells are broken. And these kids, how they show up? They show up because they're in the pediatric office all the time. Another infection. You got a fever and you don't know what to do. And to a certain extent, kids in the first year of life, especially if they go to daycare and they're exposed to all kinds of stuff that they've never seen before, they're sick a lot. But these kids are even sicker than that. So that's the earliest sign that we need to work very well. These kids don't just have the regular, viral cold stuff that you expect. They start to get infections with things that don't usually cause babies to associate with sick. They're sick with oxbrosis and tick infection. Usually after a baby is more than a month, 6 weeks old, they don't have problems with Candida anymore. They don't have thrush anymore. But these babies keep having, infections that the immune system is typically able to handle. But you're seeing the same kind of things over and over and over. And that might make you think you check this out. What do? You got to keep now there's some other fancy stuff that we can try to do. Stem cell grafts and other things like that. And doing therapy was tried and was failure. I think they tried it in France, and the kids who received it was a viral vector that they put into try to get the proper genes into the trial cell line. Kids seemed to be better for a while, but the kids developed a life-threatening leukemia, so they had to pull it. We'll get there when we go through the same time. And we talked about secondary warning sites, cancer, diabetes, therapy. That is recognized infection that causes deficiency, but other infections, if you've got a person with leprosy, they're going to be in the same kind of thing. Their immune system is working so hard with the infection that there's just comes to other things. People who are on corticosteroids chronically, like that SLE patient that we were talking about, are going to be more susceptible to infections with corticosteroids. Why? Put your immune system settles down for a nap, so that infections can come in and cause. Cancer, is some uncontrolled and abnormal proliferation of cells. The cells are growing in a very disorganized manner, and they don't have the usual. They don't respond to the usual signals that the rest of our body has to say, okay, that's enough cells, stop making more cells. Like our liver knows that, okay, if I have, say, hepatitis, and I have some damage to the current liver cells in my, and I start to get better, and my liver starts to produce healthy cells again. There are signals in my liver that tells it, okay, stop, that's enough cells. The cancerous growth they don't pay attention to those types of signals, and it keeps growing until matches are very big. You know that benign was a non-cancer. And this is kind of what I alluded to when I said if we live long enough, he will probably at some point develop a cancer because our immune system over time gets less capable of noticing when cells are not doing something right. When there is a virus in my cell that tells my cells that it changes the antigens on the surface, our cytotoxic T cells are going to see them and get rid of them. So they're not going to be allowed to grow and grow and grow and grow. But with age and the inability to respond to those kinds of changes on the surface, cells will be able to grow and grow and grow, and our immune system will be able to check it. That's why we're not walking around with growth and nodules and stuff all over our bodies, because our immune system keeps them in check. That's why people with HIV/AIDS have a sleepy immune system. T cells that don't work clean as they can, and they want to stay away from pesticides and all kinds of drugs and medications, anything you don't need, you don't want, because you don't know which thing in what situation in what environment might be movement and we have too much recessive genes in our genome, but again, they become less able to do the work that they were designed to when you have all kinds of things on board, medications, radiation. Now this is the protocol disease. These are genes that are in our genome that regulate mitosis during normal growth. They can say, okay, that's good, that's enough. It can override, control cell division, and one of the theories of why some people are prone to cancer, some people get cancer, is because their ability to regulate mitosis is more, or somehow those genes have been activated. And it shows a switch that tells cells when to turn on, when to turn off, when to rest, when to divide, and when it's not working properly, the oxygen is not working properly, there's no signal to stop. There's no rest signal. They just divide. And you end up with this big out of control mass that affects other parts of the body. Viruses, we keep talking about viruses and their ability to transform normal host cells into cancer cells. Sometimes the virus genome itself, and we'll talk about some of those, we'll talk about the virology, can be inserted into the DNA of the host and take over the host control of cell growth. The human papillomavirus causes cervical cancer, so now we have a vaccine that's called what? Gardasil, to work against that virus, so that the virus can't cause cervical cancer. Epstein-Barr virus is the virus that is implicated in Burkitt lymphoma. What else is Epstein-Barr virus? Not MS.
Mono.
MS, but what?
Mono.
Mononucleosis.
Does that mean someone who gets mono has a higher propensity to get Burkitt lymphoma?
No, well, it depends. The people who get Burkitt lymphoma, it's rare that you're going to see it in the United States. These are people who it's more third world-ish, and it could be because their immune system is already not just because of nutritional, environmental, and access to care kind of thing. But a regular, ordinary person in a first world country with mono is going to be 6 to 7 weeks and they're not going to do it. If you get Burkitt lymphoma and you do the viral test, this is another cartoon that says instead of lysing the whole cell, going and doing what the thing, viruses do. You know, viruses can infect cells, they grow and grow and grow, they lyse the whole cell, and then they go and infect other cells. Sometimes, instead of that, they become latent. They insert themselves in the whole genome, DNA, and doesn't like it, but it's transformed into an abnormal cell. And that abnormal cell can be a cancer cell. So that's why people will sometimes say, and I will sometimes say, that cancer is an infectious disease. Because if you think of it like this, you can see why it follows the logic of why someone might say it. Some cancers are associated with chromosomal traits, and we talk about that as mutations, alterations in the normal DNA. And this is that some translocations within the convergence of ribosomes with leukemia, diseases, and I'm not going to ask you, say, mention melanoma and have you tell me that it's ribosome. I just need you to know that these are some of the kinds of things that can result in cancer, the kinds of changes in DNA can result in cancer. The first bullet point is kind of already talked about. It's the reason we don't walk around with cancer, tumors, and things hanging off our heads. It's because our immune system will discover the minute changes in antigens on the surface quickly and just get rid of them. So they can't continue to just take our bodies over. That's what our immune system does for us every day. Cell-mediated immunity, okay, those guys who can't make cytotoxic T cells, of course, they're going to be more at risk for cancer. Immune system failed in cancer. Why? The first little circular bullet point says that cancer cells may not be immunogenic enough. We learned about immunogenicity. And so if a cancer cell is not immunogenic enough, what does that imply? Well, what you already know. It doesn't have anything to do with cancer. If cancer cells are not immunogenic enough,
They don't fight it off. Your body doesn't fight it off.
why?
Doesn't recognize.
It doesn't recognize. It's not obvious enough to the immune system that this is non-cell. Remember we said big changes, big whole cells, complex antigens are the most immunogenic because it's easier for the immune system to be able to respond to it. So if cancer cells aren't immunogenic enough, the immune system goes, I don't see anything wrong with that. Why should I be getting upset about that? Why destroy that? It looks just like everything else around me. So that's one theory of how an immune system fails. Cells may retain self-markers. So it's kind of saying the same thing. The self-markers are more prominent to the immune system than the cancer markers. So they're not going to be targeted for why the transient failure of the immune system. The immune system could be speaking with somebody else. There's something else like we're talking about in third world countries. The immune system is just overwhelmed with GI ailments or something. You could have a belly full of worms, and the immune system is too busy to be able to notice it's only an virus. It's not a big deal. We can take care of that. So
Remember, I'm recording.
Okay. So you're doing Clin Lab stuff, so this kind of stuff you're probably starting to feel more comfortable with. So you're doing lab, and you figure stuff out in the laboratory, and you figure out what the person has and what's going wrong in the lab. Well, some of the stuff we've already talked about. You know about the rapid morphology of the cells. Sometimes you can look at them and kind of figure out, kind of know what's going on. You can look at how they grow on solid media, kind of know what they might be dealing with. You can take pure cultures of an organism and put them through different biochemical tests, see what they grow on, what enzymes they make, and kind of figure out what the organism is that you're dealing with. Most often when you send a specimen for identification and sensitivity, they'll do all these things for you. They'll tell you what organism you're dealing with and what antibiotics will not work. They will tell you which ones to use necessarily, but they'll get you moving in the right direction. And we're getting better and better with these things. We're able to send a specimen out instantaneously. They'll touch something down on a probe and tell you by the genome of the organism what you're familiar with. Antibodies you can detect antigen and figure out what the next person is that. How to collect specimens, handle them, transport them, store them. If you work in a hospital, you don't have to worry. You do what the lab tells you to do. If you do a test for, say, diphtheria, they'll give you a coffee swab. They'll give you swab, they'll give you but if you're out in the field somewhere, you'll have to be more knowledgeable and strict about what you culture things on, whether you keep it cool, what temperature it has to be, how long you think it takes before you get to the lab, and those kinds of things that impact the likelihood of the lab activity. Something for you. These are all the places you can culture from, and that is a common little rate, little if you got media in it, you got something to swab the site with. This is in a this is like a full class. And once you put the specimen in, break it so you have fresh sterile media to sort of bathe in your swab and so you get to it going. It's sterile when you open it, so it's a great thing to just add in your work for many things, doesn't work for everything. And it kind of tells you that you can do that. With blood cultures, you're going to have a strict attention to how you prepare the skin before you put a needle in it, how to select anaerobic cultures because you don't want them to, you want to kill it by having it exposed to a lot of air before you get it to the lab, how to get it there any types in this? That's really pretty easy to do. You need a wet prep, you need some KOH, a slide, a cover slip, and something to swab or whatever it is. And you can look at it, so you know what you need to do. Scalp wounds, skin wounds, urine, or whatever, it's direct, and you're ready to roll. Immunologic means you're typically taking a serum substance, a serum test, and you're looking for antibodies. And sometimes you need, like on the test, and most people are pretty good on this, the one that shows titers, and you are looking for an increase in titers to understand that there's a infection now, or that they have been exposed at some point, and they've had a relatively high titer of that antibody in the blood. Sometimes one sample isn't enough to do an immunologic test. You can do it over time. Culturate, all about that. Two categories of results. We're pretty sure what we got. You see under the microscope that it's gram-positive hot-sided change. And you kind of know. We're going to send it to the lab anyway to know if we're dealing with group A hemolytic stress, so we know we have to continue for just to get the next week. Some other ways we can do direct testing. All kinds of ways we can do biochemical, serological, and real fancy stuff. We talked about what happened testing at some point in our teaching. And then the patient who can take blood and do immunologic type test. So next again, you just look at it, kind of know. This is an answer test where you really do know. It's not presumptive. This is pretty confirmatory. Syphilis is really, really, really hard to stain. It takes gram stain syphilis and expect to be able to identify whether you have it or not. But you can do this fluorescent stain when you take your specimen and you fix it to the slide, and then you add to your specimen an antibody against syphilis. But the antibody has a fluorescent dye attached to it. So if there's any syphilis on the slide, the antibody is going to do a handstand on the syphilis. And so it's not going to get washed off. It's going to be stuck on the specimen. And you look under a fluorescent microscope, and if there was syphilis there, they did a handstand on it, you'll be able to see the syphilis, the spirochetes glow, kind of like that. If there were no syphilis organisms on the slide, the antibodies would go on, but it wouldn't find anybody to do a handstand on, so you wash it and it will wash right off. And you wouldn't see any glowing organisms in the rest of the life. So that's the antigen, antibodies, and fluorescent testing to know what you're dealing with right away. Culturing takes longer. Biochemical test, that takes longer. You gotta wait to see what enzymes are present, how it changes the color of the media, what carbohydrates that the organism is metabolizing. But we have done something like this in microbes. We have an genome and inoculated and all these different kinds of media, incubated and wait. And what the organism does with all of these substances. And depending on what happens, you go look on a chart and they'll say, well, if this one is clear, if this one is dark yellow, and this is yellow, and all this, then that organism is well. We did something similar. All right, this was just too crazy to try and explain in a minute. We'll use maybe 2 minutes. So we'll start here.